Um, Chapter 20 is how we actually have, first of all, how we know what the genetic code is and how we manipulate genes nowadays. We can actually go in and make changes to genomes. All of that is molecular genetics. So this is where we really get heavy duty into molecular genetics and the tools of molecular genetics. I like this little picture because that's essentially what we're doing is slicing DNA. We don't use scissors, but pretty close. And I'm going to show you what we do use and how that all hangs together here in just a moment. Common tools. One of the big tools that we use in molecular genetics is recombinant DNA. You make a recombinant organism. This is a picture of a bacterium, and over here, where my pointer is right now, that's its standard DNA that just kind of flops around the cell because it's a prokaryote. It doesn't have a nucleus. These are plasmids. And plasmids are little additional bits of DNA that are often present in bacteria. They'd be present in normal bacteria even if we hadn't introduced them. What they're starting to do now, or what they've been doing actually for quite a while, is you can manufacture a plasmid. We can insert genes that we make, and then we can stick them into this plasmid, and then stick the plasmid into the bacteria. The bacteria, the cellular machinery in the bacteria is stupid. It'll read whatever DNA is there. So it reads its own normal DNA, just like it does on an everyday basis. But then it's also, its RNA polymerase is going to go in there and it's going to read the plasmid as well and produce whatever protein we've given it instructions to build. And that's good for making medications. It can be good for genetically engineering bacteria that do jobs we want them to do. Like making ethanol, for example, is increasing the rate at which they can make ethanol for us, or making certain drugs. A lot of that's done through recombinant DNA. This is a picture of a plasmid down here at the bottom here. All these little abbreviations here are either names of genes or names of tools that we use to cut the plasmid right there at the place where we wanted it cut. We're going to talk a little bit about their structure in class. Restriction enzymes, these are our scissors. This is DNA scissors. What you can think of when you think restriction enzymes, it's an enzyme. It's a protein that speeds up chemical reactions. In this case, we're going to be breaking chemical bonds. And the chemical bonds we break are going to be the sugar phosphate bonds in the backbone of the DNA there and there. And it breaks, this particular enzyme is going to be sensitive to one particular code. So for example, this particular restriction enzyme is a restriction enzyme that cuts only at the code AATT. Okay, that's the only place where this restriction enzyme will ever cut. So if I take an entire DNA strand from an organism and treat it with this restriction enzyme, anytime the code AATT happens on that strand of DNA, it's going to get sliced. So we're going to slice up our DNA into all kinds of little slices, but it's only going to cut where it sees AATT. That's what restriction enzymes do. You can order different restriction enzymes. You literally can order them out of a catalog now, and for codes that you want to slice at. What you end up with is, if we're taking a bacterial plasmid, which remember is just DNA, and I treat it with a restriction enzyme, it will cut at one specific region, and what it leaves is these hanging ends, or sometimes they call them sticky ends. And what that means is when I go to engineer my gene that I want to stick into this bacteria, I just engineer whatever gene I want in here, and then I make sure that the ending lines up with my, I make sure that the ending here would have the code TTAA at the very end of this hanging area, and the opposite at this hanging area, so that when I throw this plasmid into a solution and I throw my engineered portion in there, when they come together they'll be chemically attracted to each other and they'll stick, and I'll make my recombinant plasmid that has my engineered gene now stuck in with the normal bacterial plasmid that I can then stick in the bacteria, and it will just merrily read it, and voila, I've introduced a gene that didn't exist before in that critter. PCR, think copy machine. When we talk about PCR, PCR stands for polymerase chain reaction. We have already talked about DNA as a self-replicating molecule. All you need to do is get it to unzip. It'll stick in its own As long as you give it nucleotides, it'll stick in new DNA nucleotides on either side. You'll end up with 2 brand new strands of DNA. You also need to have the enzymes necessary for DNA replication. But you just take a mixture of DNA that you want replicated. Here we go. The DNA you want replicated. You heat it up briefly so that it will split apart. You stick in some DNA primers and some enzymes and nucleotides, and it'll build exact copies of that original DNA strand that you had. So if you've just engineered a chunk of DNA and you want a gajillion copies of it, this is what you do. You do this PCR procedure. They sell machines that do the whole thing for you, and you basically input your little DNA sample into the machine. You input the primers and the chemicals that you need, the primers and the enzymes that you need, and you set the machine and you walk away, and it does the whole PCR process for you, and you end up with millions of copies of your gene of interest. Crime scene investigators, when they pick up a little teeny tiny dab of blood, and they only get a little teeny tiny itty bitty little bit of DNA out of that, the first thing they do is do PCR so they get 8 million copies of their suspect's DNA. So then they can do all kinds of genetic testing, and but the first step is always PCR so that you can amplify that DNA. You can take the one little strand you got and end up with millions of strands. Exponential growth type thing. Every time you do another PCR, there's a thrilling lab that we could do. We won't. But you take DNA and you heat it up and you cool it down. You heat it up and you cool it down in the broth with all the primers and the nucleotides, and you end up doing PCR, but you do it by hand instead of just letting a PCR machine do it for you. It's really boring, but you end up with a lot of DNA from a little teeny tiny starting point, and that's because it's exponential how fast you start reproducing the DNA. Gel electrophoresis is another technique used in molecular biology. Basically, you take a sample of DNA, you treat it with a restriction enzyme. These are all names of restriction enzymes up here that this sample was treated with. And then once we've done that, we've sliced up the DNA into all different sizes. And then we take our sample of DNA and you squirt it into these little chambers down here. Each one is the same DNA, but it's been treated with a different restriction enzyme, so it's been sliced in different places. And you end up with different sized chunks of DNA. And by doing that, then you load it into these little wells that are cut down into this gel. You run an electrical current through the gel, and the electrical current's actually going to pull the DNA through the gel. But if you could imagine this at the molecular level, the DNA, it's like it's trying to run through a forest or run through a thicket of brush or something. If you're a big, huge chunk of DNA, you're going to have a hard time running through the gel. If you're a little teeny tiny bit of DNA, you'll have an easy time going through the gel. And that's what electrophoresis is all about. The little chunks are going to travel far. So if you look at these right here, this restriction enzyme, he cut the DNA only once, so you ended up with like 2 big strands. Okay, those big strands can't run very far through the thicket. You run it for a set amount of time, they don't travel very far. If I'm a little teeny tiny strand, like right here when I cut with this restriction enzyme on my DNA strand, I cut with this restriction enzyme, it sliced it up into lots of little teeny tiny parts, and one of the little teeny tiny parts ran that far. The other one could run that far because it was even smaller. The further you run, the smaller chunk you must be. So that's how we use electrophoresis. You can actually measure the size of the strands. What this does is, since we know the genetic code where these are going to cut, and we can tell what size strand we get, you start to piece together what genetic codes, what must be present where on the DNA, and how big the strands are. And these are some of the jumping off steps for doing things like the Human Genome Project, where they figured out where every single little A and T and C and G is on the entire DNA strand. All of this technology leads to this DNA microarray chip. Basically what this is we get little snippets of human DNA, and then we're going to take your DNA and compare it to the little snippets that we have by, they line up the DNA onto the chip, and then they actually have the pieces of DNA that come in and line up, and the nitrogen bases will actually link together. The ones on the chip have a radioactive marker on there that basically is activated when the appropriate gene shows up. So if you match, the genes on the chip are like standard human DNA. If you match standard human DNA, that little blip on the chip lights up. If you don't match, if you're different, if you have a mutant genotype for that particular gene, you don't light up. So in that way we can tell where you in particular differ from the rest of the human genome And that can give us clues about what sort of genetic makeup you have, what diseases you might be prone to. And in the very near future here, people will carry around a gene chip that will have your genetics on it. You put it into a scanner, it'll compare your genetics to standard human genetics. And in that way, we could tailor medicine to people. It's going to revolutionize medicine. It's a pretty big deal. So that is it for molecular genetics. That's the rundown of what we're going to cover in class. Hope it was helpful. That was my first shot at this, so we'll see if it's any good.